Today we are going to be talking about E. coli 0157 and E. coli 0157H7 agglutination testing. And the kit we're going to be using will be the Remel E. coli 0157H7 latex kit. There are different manufacturers of this test, but we're going to go ahead and use the Remel latex test. The first part of this presentation is about testing information, and then the last part will show you how to actually perform the testing. So E. coli 0157 H7 may produce a toxin which causes hemorrhagic colitis, which is also called EHEC, or enterohemorrhagic E. coli, and also hemolytic uremic syndrome. The E. coli version of the toxin was named virotoxin at first because of its ability to kill viro cells, which are African green monkey kidney cells in culture, which is when they first discovered this and started testing for it. Shortly after that, though, the virotoxin was referred to as Shiga-like toxin because of its similarities to the Shiga toxin, which is produced by Shigella. So now, Shiga-like toxin and Shiga toxin are pretty much used interchangeably, although the actual Shiga toxin is produced by Shigella. So STEC, which is Shiga toxin producing E. coli, virocytotoxic E. coli, which is VTEC, and enterohemorrhagic E. coli, EHEC, are basically all interchangeable as well. It's kind of all means the same thing now. So you want to perform this test from isolated clear colonies on MAC with sorbitol or SMAC. These are non-sorbitol fermenting. Sorbitol fermentation is an extremely reliable way to differentiate 0157 from non-0157 E. colis. E. coli serotypes other than 0157H7 can also produce virocytotoxins, but they're usually not associated with the bloody diarrhea, the enterohemorrhagic E. coli. Don't forget, though, that this is important. If you have clear on McConkey with sorbitol and you do your 0157H7 testing and it's positive, you have to perform an API 20E or other identification to confirm that it's even E. coli. There may be other genus and species that are clear on McConkey with sorbitol and may cause a positive on this test, so you have to prove that it is actually E. coli. So do an API or whatever identification test your lab does. If you're looking for Shiga toxins only and not the 0157H7 antigens, Shiga toxin testing is performed separately. Remember, this agglutination testing only confirms the 0157 and the H7 antigens. It does not confirm toxin production. In order to determine if your specimen produces Shiga toxin, you have to do a separate test. So we're not doing Shiga toxins. We're only testing for 0157 and 0157H7. Again, if you have an E. coli 0157 or 0157H7, it is not necessarily a Shiga toxin producer, though. You can have the 0157H7 and it doesn't produce Shiga toxins. But also, if you have a Shiga toxin producer, so say you do the Shiga toxin testing and it's positive, it's not necessarily E. coli 0157H7. For example, E. coli 026 may also produce Shiga toxins. So Shiga toxin production and E. coli 0157H7 antigen testing antigens are not mutually, they don't go together all the time. You can have one and not the other. If you have enterohemorrhagic E. coli, the 0157H or 0157H7, it will make you sick. But having EHEC, which produces Shiga toxin, will just make you sicker. So that's the deal with that. So here is the kit for the 0157H7, the Remel. You'll see here there's a white top. These are all latex reagents. Here's a white top, a blue top, and a green top, and these are all latex reagents. The white top here is a non-0157 latex. This is the control, what they call the control latex. It has no 0157 or H7 antibodies attached to the latex beads. The green top contains only 0157 antibodies on the latex beads, and the blue top contains only H7 antibodies on the latex beads. Okay, so you can't test for both of them at the same time. You have to test for 0157 and then the H7. So now we're talking about quality control reagents. This is the positive and negative controls. The red top here, it's a smaller vial. These controls are smaller vials than the latex because you use less of them when you do testing. The red top contains both 0157 and H7 antigens, so both the green top and the blue top latex agglutination should show positive agglutination if you are mixing it with the red top, positive control. The yellow top does not contain 0157 and H7 antigens, so both the green and the blue latex, if you mix it with the yellow top, should not show agglutination. Another thing I'd like to point out is that this kit contains white plastic mixing sticks. You can't use wooden sticks for this because wooden sticks can give you a false reaction. The wood actually reacts with the reagents and may cause a false reaction. So a good rule of thumb for these agglutination testing kits is to use the sticks that are contained in the kit to mix your latex and your QC reagents or your latex and your specimen together. Don't just assume that you can use wood sticks. So just go with whatever the kit has. Okay, so the next part of this will be an actual video of how to produce or how to perform the testing. All right, thanks, you guys. Okay, so here you can see I'm setting this up. This is the way I set things up all the time for agglutination testing. You should do it however your preceptor or your lab prefers you to do it, or however you prefer to do it, but I'm just showing you how I do it. If you'll notice here, I have positive and negative, and this is for my latex reagent. My positive latex reagent, which is the green top, which is the 0157. This contains antibodies, 0157 antibodies, okay? The white top that I am putting on right now under my negative, and I always start with the negative first so I don't get cross-reactivity from the positive to the negative. I put a drop of the negative on each of the test ovals on the bottom row. Then I put on a drop of the positive latex reagent onto the top 3 ovals. And so I'll do that right now, and you'll notice that I'm holding the bottle up straight up and down. Remember, you have to hold the bottle straight up and down. See, this is the E. coli control latex, which is the negative. Hold the vial straight up and down to make sure that you have the proper amount of latex dropping onto your test ovals, because if you hold it to the side, it gives you a different volume. So always hold your vials straight up and down. Okay, so next what I'm going to do is I'm going to take the positive control reagent that has the 0157 and the H7 antigens, okay, and I'm going to put a drop. You'll see it says positive, negative, and patient. So I'm going to put the positive control reagent on the left 2 oval dots. I'm going to put the negative test control reagents that doesn't have the 0157 or H7 antigens onto 2 middle negative test ovals, and then I'm going to put my patient's specimen on these 2 drops. So you'll see that this is the positive E. coli 0157H7, which contains both antigens. And to those 2, I'm running out of drops. And I'm going to take the negative control, which does not have the antigens. This is not 0157, H7. I'm going to put them on both those in the middle. And then I'm going to take a test stick, or a mixing stick, and I'm going to mix the ovals. So always make sure when you mix that you go the whole way around the test oval. And you'll notice that I'm using the plastic white sticks that go with the kit. Good rule of thumb, always use whatever is added into the kit for your mixing. You can't use wood sticks on this because it may react and give you false results. So you always, good rule of thumb is always use whatever mixing sticks they give you in the kit. Now I'm going to take my clear on my MAC sorb, and I'm just going to take just a little bit of colonies, and I'm just going to just put a little bit on each of those ovals. So what I did was I used my stick, I grabbed a little bit, and I just put a little smear here, you can see it, and then I put a little, the rest of it on here, and I mixed this up. Okay. And I'll take another stick to avoid cross-contamination, and I mix that up. Again, using the entire test oval. It's so much easier to see when you use the test oval. So when you think about it here, and I'm mixing this hard because you have to get those antigen-antibody complexes to get together, so you have to make sure that it's completely mixed together. So rock that good. And so you have a positive and a positive here, so this should agglutinate. This negative latex reagent doesn't have the antibodies that you're looking of the antigens you're looking for. So this bottom row should always be negative. If it's positive, you have a problem and need to work this up further. And there's a whole big thing in the product insert. Here's the negative that does not have the antigens, so this should not agglutinate. This positive, I don't know if you can see it here. It's starting to get sandy. And then you also check your patient positive to see if that also agglutinates. This is kind of hard to see. This isn't a very good picture, but you can see here how it's kind of getting sandy. So there's your positive. So you know that your test reagent is working the way it should. And then so you can compare that with your patient sample to see if that's positive or negative as well. All right. Thank you.